great to see you all. My name's Paul Stewart. I'm the Dean of Medicine and the Dean of Faculty of Medicine and Health. And it's a great pleasure to welcome you all to this afternoon's session. What I'd like to do in the next 10-15 minutes or so is give three main messages. One, that it's important that you think about supporting cancer research. Two, to give you an overlay of the distinctiveness of what we offer in Leeds that I think is unique, our unique selling point, if you like. And then thirdly, as we build this up with John and Susan later, is pinch points in the current system where you can have the greatest impact. So our vision is very simple. It's scale and simplicity of biomedicine and health across Leeds, a top 10 university with, I see Michael Arthur's in the audience. There's only one hospital that's bigger than Leeds Teaching Hospital, and that's UCH, but it's only by 5 beds. So we're just putting another 6 in Leeds as we speak. But seriously, we are one of the biggest, second biggest teaching hospital in the UK, and that combined power of world-leading teaching hospital with a top university should mean that we're a biomedical campus of excellence. And we've some way to go, but we're well on the way. Cancer is a key platform of that. I made no apologies for showing that first slide, and there was a 7-year-old boy you might have seen in the bottom right-hand corner of that. And that drives a very important message home to me, something has been ingrained with me for many years. As a medical student in Edinburgh at the Children's Hospital, I had the misfortune to see the very, very first patient I'd ever see die. And it was a 7-year-old boy with leukemia. I've only been in medicine 30 years, but now that same child has got a normal life expectancy. So I'm convinced that in my remaining time in medicine, I will see a cure for all cancer. That seems a far reach, but I'm pretty certain it will happen. It's a pretty important diagnosis. 1 in 3 of us are going to get it. It's 37,000 new cases a year in Yorkshire, 1/3 new cases annually. And we know that there's disparities in care. Incidences of cancer vary up and down the land. In fact, Yorkshire is not great, and it's even worse in the cities in Yorkshire, Hull being the black spot, then Leeds, than it is if you live in North Allerton. And there's various reasons we might talk about for that. But on that background, as I say, there have been huge gains, and many of us now regard it as a chronic disease. But there are real examples, and Susan's going to illustrate that with her work on brain cancer, where we really still have much needed work to do. So to put this in scale for where it is for us, it's almost about 20-25% of what we do in the medical school. It's 200 academic staff. There's live research income in excess of about 50 million, and we've got various badges of accolades: Cancer Research UK, Yorkshire Cancer Research, MRC programmes, European networks. And the big thing is this is the Bexley wing. This is a fantastic hospital. And again, many of you may not know, but this is actually the 2nd biggest cancer centre in Europe. So the whole branding of this and projection is what we've got to do. And within it, there are some world-leading scientists, and I showed just a few here. This is Phil Quirk, who's pioneering new pathological techniques in colorectal cancer. Julia Newton-Bishop has discovered new pathways, new genes that cause melanoma. Peter Selby, you may recognise, has really pioneered many local, regional and national cancer networks. And you heard from David Sebag Montefiore last year talking about some of his pioneering radiotherapy platforms. And it's that real desire to spur the next generation of leaders in cancer that's part of all the effort we make in terms of that nurture from within. And you've heard from Saman, I think, and Nick this morning. We pay a huge amount of attention to grooming the next Peter Selbys and Julia's. I'm very proud of this picture. This is actually the majority of our clinical academic trainees, our junior clinicians coming through, which will be those next world leaders in cancer. This is actually an event we have every term at Wood Hall. And you'll see how Sir Alan Langlands and the university are supporting this pipeline through unprecedented investment. But your help in this space makes a huge impact. So what I'd like to do really in terms of where the Leeds USP is just pick up on 2 themes. one being what's called precision medicine and the other being informatics. The precision medicine is really described in this somewhat complex slide, but it basically summarises the in-house discovery, the very essence of new discovery medicine, new pathways, new genetic causes of disease, from which we can build on that. And through this escalator, which you may have heard about in the press, takes 17 years, our vision is to do this in 17 months. To take that very early discovery, to put it very quickly into humans through clinical research facilities, to go through very quickly the evaluation of these. Does it work? Importantly, can we afford it? And then very quickly at the other end, to start turning that round to improve the health of the local, regional and national population. And that in the essence is precision medicine. It's the ability to give the right medicine to the right patient at the right time. And it's complex. These are the people we have to work with on a daily basis in order to pursue that engine. Key partners, all of this for us has just come together in Leeds. We've been largely on the back of this track record, awarded an Innovate UK Precision Medicine Catapult Centre, largely because we can bring all of these partners together to deliver that gain in human health. Now for cancer, I wanted to show that with an illustrative case, which is from some of our world-leading hemato-oncologists. This is a common form of leukemia, a leukemia of white cells, the lymphocytic leukemia, and this is a patient presenting with huge amount of malignant lymph nodes, in this case in the axilla. And the precision bit comes in here, because we can take cells from this individual, be it his bone marrow, bloodstream, or the lymph node itself, and we can do clever things back in the laboratory of that, with that, to determine why that particular individual has got that cancer. And in this case, when we do that, we turn out to find out that he has a defect in a pathway. It doesn't particularly matter what that pathway is, but it's a specific pathway involving a kinase enzyme in DNA repair. And that means we can then target a new biological. Again, the bulk of this work was done in Leeds in terms of the investigation of this. This is a new compound called ibrutinib. And you see in this patient, this is a scan showing this patient's abdomen effectively full of tumour. And then within just a few weeks of this therapy, it's all gone. So this you see is that now you can see the normal kidneys, this is the liver, this is the spleen. Dramatic difference. So this patient, through that personalised approach, has been cured of his leukemia. This is another patient, again with a slightly odd malignant form of a hematopoietic disorder. This is actually one of my patients. I'm an endocrinologist, but he turns out to have some weird and wonderful hormone problems as a result of his cancer. But he presents with breathlessness and weight loss, not surprisingly, because he's got literally kilograms of cancer tissue in his chest. This is a PET scan showing the cancer tissue. Again, we take that tissue, we find that he has a particular mutation in a pathway, in this so-called BRAF mutation, and with targeted therapy against that pathway, then you see a dramatic improvement 90 days later on the scan. So it's that personalised approach which is so key, and is a real distinctiveness of what we do in Leeds. Now, the other bit that I'm sure hasn't missed your attention is Leeds as the data city of the UK. And that's come together through an Institute of Data Analytics, and a big part of that is an MRC centre in medical bioinformatics. This is our GCHQ. It's on top of the Wordsley building. It's 42,000 square feet of data informatics. And I just want to bring that alive to you. This was taken just a few days after it opened, just actually 2 or 3 months ago. And this is how we use the data that's very powerful in that sector. Jeff Hall, one of our cancer gurus, has that kind of informatics brain. And what Jeff has largely done off his own back is do something which you probably saw in the recent press Adam Brooks Hospital has failed to do, which is combine primary care, secondary care and social care data, all in a single point of entry. So this is the so-called Leeds Care Record that spans the Leeds Teaching Hospital, virtually every GP practice. So at any one time we have live access of nearly 500,000 case records. That's the Leeds Care Record. And what Jeff's been able to do with that electronic patient record is this is what he sees on his desktop. Let me just talk you through it. Because we've been able to wire that through to Health and Social Care Innovation Centre, hospital episode events, national statistics, what Jeff has here is columns of outcome data. This is mortality at 30 days right through to 5 years. National mortality data that's set from midnight the day before. So this is live outcome data that he has on his desktop when he then is looking at a whole host of patients, be it with breast cancer, gastrointestinal, lung, or gynecological malignancies. This is the case set that we see in Leeds, and these are the numbers of patients that have died in our service. But faced with the individual patient in front of him, he's able to give them real-time outcome data, which doesn't happen anywhere else in the UK. But the other thing Jeff's able to do is to go in and interrogate those data sets, not on an anecdotal level, but now, as you see, in 100,000 cancer patients. And based on the anecdote that Jeff felt that some of his patients with diabetes weren't doing as well, he's been able to look at that, as I say, in 100,000 cases in terms of those that had diabetes at the diagnosis of cancer versus those that didn't. And hey-ho, if you've got diabetes and cancer, your mortality is roughly doubled. But Jeff didn't stop there. He looked at the differences across cancers, and it didn't really matter for lung cancer. Diabetes had no impact if you had lung cancer, but if you had melanoma, it was a devastating effect. So we're now going backwards. By interrogating that data, Jeff's looking at what the underlying pathways are that make diabetes such a dreadful diagnosis if you've got melanoma, but don't matter a snitch if you've got lung cancer. So this is really what it's all about. This is how we're coming around to impact on patients. This is another example of a malignant form of anemia which has a 5-year survival of 50%, until Peter Hillman really pioneered this new targeted therapy. Again, it's targeted to these specific patients, and not only are they alive, but they're now having children, something that was unheard of with this diagnosis. So I'm going to stop there, but bring it back to introduce John to that first part of this precision medicine pathway, the discovery science, the inbuilt discovery that's going on in Leeds that we need a help and a jolt to get it to the next level. Funding bodies are very conservative. They like to see almost the work done, so they're backing winners before they'll put serious money behind it. As you see from here, we've got lots and lots of projects in this space that are ripe to go to the next stage, that just with that little bit of pump priming we'll get there. And I'm going to pass over to John Ladbury now, who's going to take the story forward. John.
My name is John Ladbury, and I'm the Dean for the Faculty of Biological Sciences, and as Paul has already alluded to, the Faculty of Medicine and Health and the Faculty of Biological Sciences are working very closely in trying to sort of form an interface whereby we can work in many areas using clinical activity and basic science to develop novel therapeutics. And I wanted to sort of start by sort of setting the scene as far as what we're trying to get over this afternoon. And I think Paul put this in a good way at the front end here, in as much as the people in Paul's faculty generate an awful lot of clinical data, and fantastic clinical data. In fact, Leeds is clearly one of the best places in the world, and certainly within Europe, for developing data, which includes genomic screening, includes fantastic pathological data sets, and basically an understanding of what cancer looks like in terms of what we can read from patients. What we want to do, obviously, is develop therapeutics, and that means not only drug inhibitors, but also looking at ways of developing biomarkers and biologics. But there's clearly a path in between those two, and the major issue which sort of stands in the way of getting from A to B is a biological understanding, and that's where I think my faculty come in and the work we do in basic science. And so what we really need to do is, if you understand, or if you get data from patients, you understand something about the genomics of disease, if you understand those point mutations that Paul mentioned in pathways which lead to cases of different forms of cancer, what we need to then understand is how those translate into a basic biological mechanism. What happens in a cell which makes the cell turn from a normal cell into a malignant state, whereby it's telling itself to keep dividing and dividing, which essentially is what cancer is. And so what we do is look at the understanding of these mutations and patient data and look at it in terms of how particular molecules that the genomic data throws up behave and give rise to these aberrant signaling pathways. So to sort of underline the problem here, what we have is fantastic access to high quality clinical data, as we've already seen. But there's a bottleneck in getting from those data through to a biological understanding and being able to develop the therapeutics from that biological understanding. And so, with respect to the problem, obviously I'm going to try and tell you how we want to solve that particular issue. And as I say, we can start with a huge amount of fantastic clinical data from the hospitals that we're affiliated with. We can then use those data from the genetic information, we can then produce the protein which the genetic information are encoded for. We can make those proteins and then we can put them through a structural biological program using largely three different methods. And I should point out at this point we have had huge investment within the university, particularly in NMR and electron microscopy, and combining with our already existing capability with crystallography. We are one of only 6 institutions in the whole of Europe which have state-of-the-art capability in all 3 areas. And armed with these techniques, we can take protein and produce a structure which allows us to look at the molecule that we're trying to target to generate our therapeutics. We can then take that structure information and the protein that we've generated at this early stage and look at and disseminate it and provide a biological understanding of how the molecule is involved in giving rise to aberrant signaling, which leads to the oncogenic state, as I've already said. We can use those the proteins to screen for small molecules, which will then allow us to develop inhibitors, either using actual libraries of molecules or using computer-based methods. And we can also derive biophysical data, which helps us to underpin how the potential drugs and how efficacious they may well be in terms of inhibiting the process which gives rise to cell proliferation. And of course, this central capability leads into that final stage, which is where we want to be, which is developing therapeutics, i.e. drugs, inhibitors of aberrant signaling pathways, biomarkers which can help clinicians make decisions on patient outcome and what therapeutics will be appropriate, and leading into clinical trials, which again leads hospitals have capability which is second to none in many areas that we're interested in. So in terms of where we are, and I just want to give you an overview of the capability that we have at the university today, we have state-of-the-art equipment to undertake the research that I'm talking about today. And you can see here, just with this sort of flavour of the capability that we have, we can cover all of these areas with state-of-the-art equipment, and I'm not going to go through all of these individually, but we have an amazing robotics screening, which I think you saw some of the capability in engineering with regard to robotics. We can apply these methods to screening capability and high throughput methodologies. As I've already mentioned, structural biology and the techniques that I highlighted, you can see some of the colossal instrumentation here. But what I'm saying here is that we have fantastic equipment at this university, and we're making significant investments in being able to translate the basic understanding from the atomic level and give a much clearer picture to provide a biological understanding. So we can sort of summarise that in terms of our research capability, and we have strengths in all of these areas, all of which provide that part of that pipeline which I showed you in an earlier slide. A biophysics capability, which again is second to none, and this integrates activity not only from my faculty but also from the schools in mathematics, mathematics, physics and chemistry. We have an incredible capability in chemical biology and medicinal chemistry. I've already mentioned our strengths in structural biology, and this is also something that we clearly need to engage with, understanding what happens to the molecules that we're interested in when we take them into cells and how they, within the cellular environment, can give rise to cancer. So we have the capability. We have great research, and this is a bit of an embarrassment now in the next couple of slides, in as much as I'm talking about great researchers, and my colleagues told me to use myself as an example, and clearly that puts me on the spot somewhat, and there are much greater researchers than I, but I will kind of give you a flavour of my background coming here from this institution, which is situated in Houston, in Texas. This pink marble edifice is MD Anderson Cancer Center, where I spent 6 years. This is the biggest cancer hospital in the world, and just to give you a little bit of a flavour of where some of my education in cancer research came from, this is a fantastic research activity, as well as a biggest cancer hospital in the world. It has 600 beds and deals with over 100,000 inpatients and 1 million outpatients per year. It ranks first in the National Cancer Institute grant income receivers in the United States, invests something like 700 million per year in cancer research, and has an endowment of something like $500 million. And this is where we're aiming for, obviously, with the University of Leeds. So in my role at MD Anderson, I was the director of this Centre for Biomolecular Structure and Function. And the reason I'm showing this is that this centre was set up when I arrived, and the remit of this centre was to do exactly what I'm trying to propose today, is to engage clinicians in basic research with a view to developing therapeutics against cancer. And the way that it worked was to provide the capability, the kind of capability I've shown you that we have already at Leeds, and induce clinicians from the hospital who are dealing with patients on a daily basis, who have a wealth of information that we can use as basic scientists, and forming that interface to allow us to work with them to develop a biological understanding to develop therapeutics. And this was a successful operation, and when I left, we had at any given time 25 projects running, which were all engaging clinicians with basic scientists and producing opportunities to develop great therapeutics. But not only that, also produce opportunities for us to write fantastic grants and to develop great papers and highly publishable research. So now I'm at Leeds. This is just kind of flavour of the kind of work that I do in my lab. We are very interested in taking this information, which is cellular images, looking at how proteins interact with cells. You can use fluorescent capability to actually look at interactions within cells, and of course these interactions that we're looking at are interactions that give rise to cell proliferative events. We can then work with our clinical colleagues to look at how these manifest themselves within in an in vivo situation. And but the key route to that is to develop an understanding on the molecular level. So we develop structures of the interactions, or structure information about the interactions that we can see in cells, and look at those on an atomic level to see how they occur, and also try and understand how we might put a compound in here to inhibit the interaction where it's causing an aberrant outcome and cancer. So I just wanted to kind of embellish some of the success stories that Paul mentioned already, and this is a story which we've been working on in my lab, and basically, and I'm going to be very brief, and I apologise, and I'd be happy to talk to you all afternoon about it if I could, but basically what we've done here is look using, again, atomic level understanding of an interaction of a protein, which is shown here in red, with a sequence of another protein, which we've sort of stripped down to an atomic stick model, if you like, and shown how that protein, those proteins interact with one another. The key feature here is that this protein, when it's bound to the receptor, which is that blue stick model, is inhibitory and keeps the receptor in a quiet and passive state. However, if you take a cell and knock down the concentration of this red protein shown here, as we've done in this fluorescent lifetime image, you'll see that another protein, which again I'm rushing through, is another protein which is known as PLC gamma, will get onto that receptor, bind instead of this molecule here, and give rise to cancer. So, with looking at this very closely, and all this work, I have to say, is underpinned by pathological samples from our clinical colleagues, you can see that in a normal tissue, this protein, if you look at the darkness of the stain, if you just kind of squint your eyes and see dark versus light, light versus dark, you can see that in normal cells, this protein is more expressed than this protein. However, in cells which give rise to, which are oncogenic, in this case lung adenocarcinoma, you can see that this protein is less expressed and this one is higher expressed. Similarly, as we've shown in an in vitro situation, there's competition. As we knock down the concentration of this protein goes up. And the important feature of that is that it's a killer. So this is what we call a Kaplan-Meier plot, and it basically shows a percentage or proportion of patient survival over a period of time, and these are months down here. And what I'm highlighting here is if we take the case where this particular protein is expressed more highly than this protein, your chances of survival are much greater than the opposite situation where that protein is low and this protein is high. And you can see here that just the change in the concentrations of those two proteins in a cell will have an effect of years on your chance of survival. So the basic understanding derived from the clinical data and mapped onto a molecular mechanism gives us an opportunity to make predictions on a patient's outcome. This is even more pronounced the previous slide was ovarian cancer. This is even more pronounced in lung adenocarcinoma, where you can see that if you have high expression of this particular protein, your chances of survival are markedly higher than the opposite situation, where these patients will survive for the most part for a normal lifespan, whereas these patients, you can see, are 50% of them are dead within 5 years. So knowing something about the expression of these proteins allows us to make observations as to how patients will survive, and also provides us with an opportunity to make therapeutic decisions which will be advantageous to the patient. The only way you can do that is by developing prognostic or predictive markers based on, in this case, the concentration of these proteins. We're working now with a company to develop these, and hopefully we will be able to use these markers when we are presented with a patient with a tumour biopsy, and we can then make a decision, in the case of ovarian cancer, for example, whether to start with surgery or go straight to a chemotherapeutic approach. And so the point here is that this work the basic biological understanding that we develop can't work without interaction with clinicians to produce this kind of data and these kind of data. And so it's absolutely important that we close that gap between clinical and basic science. But when you do, you can see how advantageous that can be. So the solution to the problem that I showed you earlier is as follows. What we need is to be able to access research personnel who will provide us with the capability at the early stages of projects to move them through that pipeline I showed you to therapeutic development. And currently, this is a major problem. The major funders in our area, in this particular area of research, usually won't back this kind of early stage research. I think as Paul said, they want to back winners, they want to know what the result's going to be. But it's very hard to know what the results are going to be when you're starting with clinical data, even though you may be armed with an amazing capability in basic science and opportunity to get biological understanding. Without the hands-on, without the people to take those data and do the experiments in cells, we really can't move forward. So how can you make a difference? Well, the fund we're trying to set up here it will enable us to pump prime early research activity, to fund researchers to take these clinical outcomes and the data from clinical outcomes to the point where we can develop a biological understanding and develop therapeutics therefrom. What does it cost? Well, the initial startup to get this thing going, which will basically underwrite a PhD student or a postdoc, is of the order of 90,000. This amounts to something like 14.7k a year for a PhD student. From this asterisk is like you find on those credit card things. With this applies to earners of a certain level, I believe. This is all it costs to engage in this process. And if we can develop a fund and which we can keep adding to, we can solve our problem. And just to underscore what Paul has already said, we are in a position where we have many projects. We have a huge resource within the hospital where there are researchers coming to me with amazing opportunities to get down that pipeline, but we can't do anything with these because we don't have the hands to take these projects forward. And I'll give you an example to finish off with. 2 weeks ago, I was sitting in my office with 4 clinicians who had done amazing work to develop and probe tumours for genes that were aberrant. They came to me with 2,000 mutations that if we could understand even a handful of those, we could make a difference in terms of understanding the disease and suggest targets which might provide opportunities for therapeutics. I couldn't move forward on those. The only way we can to move forward is to write a grant, is to hopefully get a grant, to put a PhD student in place, to then move, to take the research forward. That can put us back well over a year and more likely 2 years before that opportunity arises. And so if we had a fund where we could go to with exciting projects, with all of this capability in place, we could really move things forward. And I hate to say that the return on investment will be immense, not just in terms of financial output, but in terms of patient care and patient health. And so I'll hand over now to Susan.
Thank you. So I'm going to tell you a little bit about brain cancer and the research that we're doing here in Leeds in that context. So I'm going to start by giving you some rather scary facts, actually, about brain cancer. Give you an example of a success story that we've managed to take from the lab to the clinic here in Leeds, and then show you an example where some philanthropic investment has helped us start some new work that will tie together some of the important questions that we've found in the lab that we know are relevant to patients, and allowed us to set up a project that we'll work with John, hopefully, again, to close that loop back to the clinic. So the scary bit first. So brain cancer, as Paul alluded to, is one of the cancers that's really been left behind in the last few years, where we've all been hearing about the improved outcomes in many, many circumstances. That is still not true for patients with brain cancers. They are scary because we know that they can develop extremely quickly. And here is an example from a real patient where we saw this head scan. This is an MRI scan of a patient's head with this tiny little lesion here, which we didn't really know what it was. It was almost too small to do a biopsy and find out what it was. When we did a subsequent scan 10 weeks later, this little thing had turned into this not so little thing, and that turned out to be a very aggressive brain cancer. One of the things we know about these cancers is that what you see is not all of what you get. So what you see on the scan may suggest that this is the main bit of the tumour, but actually the harder you look, the more you see tumour cells way beyond the obvious tumour. And that's because these tumour cells are very mobile. They migrate through the brain. And there have been some very elegant data in the literature recently that demonstrate that very, very soon, after these brain tumour cells become malignant, before even there's as much of a tumour as that to see, these cells are already moving abnormally, so they're already on the march. So that makes it a very scary idea. So if we were thinking about where most of the tumour cells are, they'd certainly be several centimetres away from the obvious lump in the head. So you can see that we're looking at getting near, worrying about nearly half a brain there. This is the other scary thing about brain tumours. We don't know really what causes them. They seem to appear randomly across the population. There are no risk factors, there are no lifestyle factors at the moment that we know of, and that makes them really very difficult to understand. This is the other fact that I think a lot of people are not aware of now, and this comes back to some of the data that Paul alluded to in the leukemia world, where leukemia used to be the scariest disease in children. But actually, because of the improvements in outcome now, because of all the new treatments that have come online, leukemia is no longer a very big killer in children, but brain cancer is now the biggest cancer killer in children and young adults. So the leading cause of cancer death in anybody under 40. This is the sobering fact. So brain tumour research receives a very small percentage of cancer research funding if you look across the national portfolio from the big cancer funders. And that, of course, makes it far more of a challenge. So we're trying to address those issues in Leeds. So in the last few years, we've built up the biggest brain tumour research group that includes this happy gang in the lab, but also a big cohort of clinicians who are involved in the research. And we want to use those bright young minds and those motivated people to really try and make a difference in terms of how we understand this disease and what we can bring back to the clinic. So one of the success stories that we've been able to report in the last few years is this story. This is the model in which we use, bizarre though it sounds, we use a harmless virus that we inject into patients' blood in order to deliver an immune-activating agent to the site of the tumour. This is the virus, it's not really that big. But the idea is that this harmless virus, through the bloodstream, will home to the tumour. They get carried to the tumour actually in the white blood cells, which go to the tumour because the tumour causes inflammation. So the virus gets carried to the tumour, promotes a local inflammatory and immune reaction, and thereby kills some of the tumour cells. And some of the work that we've done in the last, just the last year or so, has demonstrated very nicely in some of our own brain tumour patients that this really might work. So one of the first studies we did was with our very good local neurosurgical team, we set up a study so that we gave patients just one injection of this particular virus just before they were due to have surgery. So that gave us the opportunity of looking at the tumour specimen when it was removed. And when we did that, we saw that the virus that we'd injected had indeed got to the tumour. We stopped the study actually a little bit early because we saw that the virus appeared in nearly all the patients that we treated. So we completed that study towards the end of 2014. Based on these data, Cancer Research UK have agreed to fund a big national study that we're going to lead here in Leeds, so that these patients with poor prognosis brain tumours will receive this injection in addition to standard treatment, so alongside radiotherapy and chemotherapy treatment, which we know are nothing like as effective as we would like them to be in these patients. And we're hoping that that study will open sometime towards the middle of next year. And this is one of the new projects that we've been working on for the last year or so. And this is to target that very migratory phenotype that I described in the brain tumour cells. So this is to try and find ways, if we can, to stop these cells moving through the brain. And here's some beautiful images of 2 brain tumour cells that look beautiful, but actually this sort of frill that you can see here around the edge of the tumour is the part of the tumour, the cell arranging the proteins so that it can move forwards. So it looks pretty, but actually this is part of the lethal aspect of these cells. So what do we know about how these cells move? Well, there are lots of different mechanisms that contribute to that, and this is one of them. This to me is a bit like cells potholing. They're crawling through the gaps, the very small gaps between normal brain cells. To do that, they need an anchorage position ahead of where they're going. They need to be able to squeeze parts of the cell, particularly the nucleus, through the very small space. And often they also then need a push from behind. And we're starting to understand which proteins in the cell contribute to those different ways that these cells move. We've come across recently a particular protein that makes a very big impact on these different aspects of cell mobility. So this protein, GSK3, we know helps the cells decide which direction they're going to move. It gives them some stability in terms of pushing in a certain direction. It helps them get traction on the material that they're moving across, and it helps them move in a consistent direction. The problem with this protein is that it's it has a lot of other effects as well, so we can't really use this protein itself to target migration specifically. Here's just an illustration of what happens to these cells if you do target this protein. I hope you can see. These are the untreated cells. These are individual glioma cells, untreated, with the nice normal arrangement of proteins, these focal adhesion points that are helping them grab onto the substrate, and these lovely tubules that give them the structure that they need to move. When you treat these cells with that protein inhibitor, they sort of collapse on themselves, really. So this cell can no longer maintain its normal structure. It's lost those adhesion points that allow it to grab onto the substrate, and it's lost those microtubule skeletal arrangements that allow it to move. So we know that targets within this protein pathway should work in terms of reducing cell motility. We are early in this project though, and as the other speakers have alluded to, it's rather difficult to get funding and to move projects like this forwards at this early stage. Happily, we've had some help, and we've had some help from this group of ladies. They're all ladies because they came together because all of their husbands died from a brain tumour. And they've been very active in terms of raising money for us, specifically to help with the brain tumour research. So they're the PPR Foundation. And in the last few years, they've helped us by funding a senior research fellow in the lab. That's the person who has done all the work that I've described to you briefly. A research nurse to help us get tissue from the clinic into the lab, and a research technician. Most recently, though, and quite excitingly, they've also agreed to fund a new PhD student in the lab. And no sooner had they agreed to give us the money than we found the student. And so this is Sophie, who's come into the lab literally in the last week to work on this migration project. The exciting thing about Sophie's project is that it's a cross-faculty project, so she will do some biology work with us in the lab, where we have access to patient material and where we've defined some of the biological questions. But the other part of the project, she's going to work with John, particularly to see if John's group and the fantastic facilities that he showed can help us to develop some specific targets that will stop these cells migrating. So a very exciting opportunity that we hope will be another success story in terms of moving from a biological understanding in the lab to an agent that we can actually give some of those patients. So we're going to finish this session then, I think, with a video. I think you will enjoy it. This is a video made by students here in Leeds, not medical students, that I think conveys very nicely the patient perspective of the sort of work that we do in the lab. I've been told to mention that it was shown at the Leeds Film Festival and has also won a prize. I hope you'll like it. It stars several members, particularly PhD students actually, from our lab.
So, anyone have? Yes.
Why is Yorkshire and Leeds in particular worse than other parts of the country for cancer rates?
Shall I have a bash at that to start with? When we arrived, I said that there's only one person amongst the three of us that doesn't really know what he's talking about, and that's me, because I'm not a cancer specialist. But it's not quite like a sociologist talking about suspension bridges. So our feeling is it's probably the comorbidities. So it's everything else that patients have when they've got the diagnosis, and that's why I specifically showed that diabetes chart. You know, so I think as we begin to understand that, it's perhaps not surprisingly, it is the fact that more people smoke in Hull, you know, than in Northallerton. It is the fact that there's a 10% incidence of diabetes for whatever reason in cancer sufferers. So we're beginning to unpick that, but it's not the fact that our patients with cancer are not getting the treatments they need, you know, in the right time, or that those treatments are inferior. I think it's the additional characteristics of the patients when they're presenting. Susan, would you?
Yeah, absolutely agree. Absolutely agree. There's nothing to suggest that there's anything particular about living in Yorkshire, but I think it's the patient populations that we find in Yorkshire that makes that impact. Yeah.
As a psychologist, let's get what might sound like a stupid question. What actually motivates a cancer cell? Why does it exist? What's it trying to do? Why does it go post-holding? What drives it?
Yeah, well, we don't know all the answers to that, but the accepted paradigm is that what drives it is a series of mutations, so mistakes in the DNA. And once there are mistakes in the DNA, the cells start behaving abnormally. And so they escape from the normal inhibitory mechanisms, actually, that would stop cells moving, because all cells could move if they want to, and or dividing uncontrollably. So that's the paradigm. But
the end result is they kill themselves, don't they? If they kill the patient, they kill themselves.
Yes, they do. But in the meantime, they basically outcompete other cells for metabolic nutrients, etc. Yeah.
Can I just add, one of the other things that we found in the research that we've been doing, part of what I presented there was not only can mutations have an effect, but it looks like fluctuations in protein concentrations have a big effect, and those don't have to be derived from mutations. They can be derived from stress factors, which we're now kind of on the threshold of opening up an area there which could have a big importance alongside an understanding of the genomic structures and so.
Good question, though. Yep.
You say that your precision medicine can get through the discovery stage in 17 months. However, have you any ideas how you can get over the regulatory mode? Which will probably take the 7 years to talk about in the first place.
And that's the bit that is the most frustrating in all of this, because I and other colleagues, I'm sure, have been on national committees, bureaucracy-busting approaches to try and get through this. And, for something where we all come together in a united NHS, the sooner we can get over the whole debacle of care.data and scaremongering, and once we enter the NHS, we all sign up for our data to be used, and we all sign up to be involved in clinical research, then we can cut through a lot of the paperwork. But we're not there yet. And for us, and Susan will testify this, to do a very simple clinical trial across different hospitals, we need repetitive paperwork to be in place across all of those different hurdles. And it is the biggest challenge, I agree, in that whole process. I think that's in the vision of the Innovate UK kind of catapult, to see if that engine can just start cutting through that bureaucracy. It's a very important point, yeah.
Tell us you're looking at what happens at the molecular and atomic level, but my brain says, well, what's happening at the subatomic level? Where does the quantum physics come into this?
When we look at the atomic level, you can always be reductionist and reduce down to the subatomic level if you want to. But I think when you start to look at the cell, and particularly when you start to look at the population of cells, which a tumour is, you have to be a little bit more holistic and look at the sort of probability of an effect occurring. So what is interesting, for example, is when you look at the kind of thing we're looking at, you can look at proteins being recruited to a receptor, and you can ask yourself, well, why does the cell, in an ideal situation, you'd only need one receptor or one protein to give rise to a signal transduction pathway. So why does the cell express in excess of 500,000 receptors and multiple millions of proteins which are involved in pathways? And it's all down to probability issues. And so you can look at things at subatomic level, but you're not going to understand how that pans out into a holistic activity. And furthermore, you can. I think with your brain, which is clearly a physics-based brain, you might want to look at it in a more statistical, probabilistic view, and that will give you a better understanding.
One last question, then we'll draw it to a close.
A simple question to Susan. Why has brain cancer historically been so stark?
Sounds like a simple question, but it's quite a long answer, probably. I think, in brief, it's a rare cancer. There's not a big surviving cohort who have much political clout, which makes it difficult. And traditionally, I think, success stories bring investment, and, we have been short of success stories, so that's made it difficult.
I think T's just through here. We're around to answer any more questions you have, and thank you for your attention. Thank you.